Thanks for joining me. This is the second in a series on the immune system, chapter 21. And in the last session, we talked about the innate immune system or innate defenses, those that are the same all the time, always in place, very fast acting. And now we're going to talk about or begin the discussion of the adaptive defenses. First, we'll learn a little background about the adaptive immune system, and then we'll talk about the humoral immunity, humoral defenses. And then in the next session, we'll follow up and talk about cellular immunity and a little bit about helper T cells, a subset of T cells that are needed for both humoral and cellular immunity to occur. Okay, the adaptive immune system. So it's a system to get infectious agents out of the body. The innate immune system essentially tries to prevent them from getting in and prevent them from colonizing some part of the body. And the adaptive immune system will go a step further in rooting them out of the body. It also serves to amplify inflammation. So there's going to be some cytokines secreted by activated cells that will ramp up inflammation. And also, there's a connection between the two adaptive and innate immune systems here that we'll talk about because, as you already know, the classical pathway of complement activation, those antimicrobial proteins, is through antibody-antigen complexes. So, of course, antibodies are the purview of adaptive defenses. The adaptive immune system requires exposure to an antigen to get started, to get things going. Priming is the word used here by our authors, and priming takes time. So there is a lag time between exposure to something foreign, some infectious agent, and a response by the adaptive defenses. I've collected together some descriptive terms to give us a beginning of a sense of what adaptive defenses are like. Firstly, they're specific. The innate immune system is a one-size-fits-all kind of a system. If anything foreign comes into the body, the same things happen each time. In this case, we're going to respond like a laser beam targeting specific individual foreign agents, bacteria or viruses or whatever that come into the body on a particular occasion. The adaptive defenses are systemic. Instead of being like a place where there's been tissue injury and bacteria have gotten into the body and inflammation starts and white blood cells come to fight the infection, this is systemic. The actors and effectors of the adaptive defenses can go pretty much anywhere in the body. Not entirely, but we'll talk about that. Memory. This may be the most important thing. Once you've been exposed to an antigen, a foreign substance that comes into the body and the adaptive immune system responds, then things are going to be different in the future. It's not going to be the same way the next time that particular agent reappears on the scene. The adaptive immune system will do something very differently. Once it's known, that agent will be attacked much more aggressively. Okay, self versus non-self recognition. In order to mount specific attacks, the innate or the adaptive defense cells have to be able to identify things that are foreign that have come into the body, and in order to do that, they need to be able to tell the difference between molecules, proteins, cells of your own body versus ones that have come in from outside and that are trying to take over your body. We'll talk about two branches or arms of adaptive defenses, humoral immunity and cellular immunity. Humoral immunity involves antibodies, the production of antibodies, which are defensive proteins that recognize and bind to any foreign substance that comes into your body, say the surface proteins on a bacterium or a virus. Those antibodies, when they bind to those antigens, they may coat the antigens and sort of block them from doing anything in your body. And they also act like tags. They mark them for phagocytosis and killing, get them out of our body, clear them away. So humoral immunity, therefore, we would say, has extracellular targets. Antibodies will be in the fluids of your body, and they can bind right to those antigens, and good things will happen. And B lymphocytes, B cells, are the ones, as we'll see later, that produce antibodies. Cellular immunity, on the other hand, involves a system that can attack and destroy cells of our own body that are infected. If there's an antigen, a bacteria, or a virus living and growing inside of a cell or a patch of cells in your body, the cellular immune system will kill those cells of your own body. It's rather kind of a dangerous concept. And this is meted out by T lymphocytes or T cells. So what exactly is an antigen? Well, an antigen is any substance, any chemical substance that can provoke an adaptive immune response. If you bring something into the body and the immune system reacts, that's an antigen. Those are the targets of the adaptive immune system, therefore. They're typically large, complex molecules like proteins. Proteins are long chains of amino acids that fold up into a complex shape, and those things are, when they're foreign, are recognized by the immune system and attacked. So proteins, polysaccharides, meaning sugar-coated proteins on the surface of microorganisms, certain types of lipids, and so forth, are our field of antigens. Haptens are interesting. They're small molecules that aren't immunogenic by themselves, but they generally cause problems because when they get into the body, they bind to proteins of our own body, maybe extracellular matrix proteins or cell surface proteins. And once they cling to those proteins, the combination looks foreign and acts like an antigen and may cause all kinds of inflammatory responses. Anybody who is susceptible to poison ivy knows what I'm talking about. This horrible, itchy, blistery kind of skin reaction that occurs because of an oil from the plant that gets into the skin, binds to some proteins in the skin, and activates a bunch of cells of the adaptive immune system that produce lots of cytokines and inflammation. So a lot of allergies, certain type of allergy response is because of haptens. An antigen is depicted here in cartoon form. This red thing in the middle represents a complex chain of amino acids all folded up into some particular shape. That's what proteins tend to do. They have secondary and tertiary and even quaternary structure. And it's a kind of complex thing. And it turns out that the immune system doesn't need that large and complex a thing to recognize. And so drawn into here is this sort of cartoonish stylization of antibodies. Those Y-shaped things are antibodies that have been produced by a B lymphocyte, part of the humoral immune system, and they have recognition sites. They're shaped like a Y, and they each have two recognition sites that can identify some chemical structure, the features of some chemical structure. And what's intended by this drawing is to show you that certain subregions within this complex protein are sufficient for recognition by antibodies or by the immune system itself, the adaptive immune system. And those specific minimal sequences that can be recognized are called antigenic determinants or epitopes. So a protein, something as complex as a protein, may have multiple antigenic determinants, and so when it comes into the body, it may trigger multiple parallel defensive processes, each of which will result in antibody production and marking and destruction. So what we're going to do probably from now on is talk as though there's a single individual response to any particular microorganism that comes in the body, but just be thinking in your own mind about the fact that there may be many, many parallel identical events taking place that help us attack and root out a particular organism from the body. There's a thing called self-antigens, and this is in a kind of unfortunate way. We're just learning about antigens, what they are, and now we're talking about self-antigens. How can it be an antigen if it's part of self? Well, it's just a term used to describe glycoprotein complexes, meaning a series of proteins that all kind of assemble together on the surface of the cells of your body. They're called major histocompatibility complex. That just means, histo means cell, compatibility. These agents are on the surface, these complexes, are on the surface of all your cells, and they're like ID tags. The cells in your body have to have MHCs on the surface that identify them as part of your own body. So that's why histocompatibility, they're compatible with your body. If they're brought into someone else's body, now they're not compatible with that body. That person has different MHCs. Everyone's MHCs are unique, and then therefore that tissue would be considered foreign. If you receive a transplant, that tissue will be foreign to your immune system because of the different MHCs that are on the protein or on the cell surface, and it will be attacked and destroyed unless we do something to intervene. So that's one thing that your MHCs do. They identify all the cells of your body as yours. MHCs also have a groove or a socket on them for holding fragments of antigens. So just like we saw the antigenic determinants on that previous slide, when macrophages or dendritic cells, phagocytes, gobble up a foreign agent that's come into the body, an antigen, we carelessly call it, it will digest that down into fragments, and all the fragments will be placed on the surface of the cell, amazingly, attached to MHC complexes. So the cell will look like this sort of laundry list of stuff that's inside this that it's gobbled up all over its surface on the MHCs, and that's important because T lymphocytes can only recognize antigens that are attached to MHC proteins. So it's a way to bring the word of a foreign invader to the attention of the adaptive immune system. So what are the cells? We've already kind of jumped ahead and talked about a little bit about cells of the adaptive immune system. Lymphocytes are the main cells of the adaptive immune system. B cells, or B lymphocytes, are the ones that are involved in producing antibodies, humoral immunity, and T lymphocytes, a certain subset of T lymphocytes, mete out cellular immunity. They're the ones that can recognize and kill cells of your body that are infected. There are multiple different subpopulations of T lymphocytes, as we'll see. Antigen-presenting cells, these are very, very important. There are phagocytic cells called antigen-presenting cells whose job it is, as we said, to display antigens on their MHCs on the surface and bring them to the attention of the immune cells, the lymphocytes, in order to trigger an immune response. So antigen-presenting cells will engulf an antigen, phagocytize it, digest it, place the fragments on its surface on MHC class 2. Major histocompatibility complexes come in two different types, class 1 and class 2. The MHC class 1, or just MHC1s, are found on all of the cells of your body, and again, those are essentially ID tags that all cells must have, or they'll be attacked and destroyed. There's MHC class 2, though, and those are specialized MHCs that are only found on antigen-presenting cells whose job it is to bring the antigens to the attention of the adaptive immune system. So MHC2s are kind of like a press pass that allows these cells to have this special privileged designation as antigen-presenting cells. So there's three types of antigen-presenting cells that we'll talk about briefly. Dendritic cells, we already know something about them. They're in the epidermis, they're in lymphoid tissues, they're sort of surrounding, like a net of them surrounds lymphoid follicles. Macrophages are antigen-presenting cells, phagocytic cells that live in many connective tissues and within lymphoid tissue as well, and in a number of organs. And B lymphocytes are actually, amazingly, antigen-presenting cells. Part of their role is going to fall into this category. Dendritic cells phagocytize antigens, digest them, or a word that's often used by immunologists, process the antigens, and place the fragments on their surface on their MHC2s, class 2. And then, interestingly, dendritic cells, if they're not already in a lymphoid tissue or organ, they will then leave the location where they phagocytosed this antigen and travel to the nearest lymphoid tissue so that they can bring the antigens to the attention of lymphocytes of the adaptive immune system. Macrophages are found in many connective tissues, areolar connective tissue. Some are found in the liver, they're found in the alveoli of the lungs, and they are the first responders oftentimes, along with dendritic cells. If a bacterium comes through the first line of defense, a macrophage will probably discover it, phagocytize it, process it, and place the antigens on its surface for the attention of the immune system. Sorry, I just glossed right over. Here's a beautiful micrograph that someone made. Micrographs, people that are good at microscopy and produce these kind of micrographs are absolutely artists, and that's just a beautiful thing. If you're not familiar with the complexity of doing microscopy, that's just an incredible presentation of a dendritic cell that someone made, and we've got the benefit of seeing that. B lymphocytes. B lymphocytes also present antigens on their surface, but B lymphocytes are different from the other two. B lymphocytes are not about the job of bringing the antigen to the attention of the immune system for the first time, their job is to get somebody to come along and help activate them. B lymphocytes, when they discover their antigen, as we'll see, they become kind of activated, but they need another cell to come along and kind of rev them up to the next level so that they can go on and produce antibodies. So that's why they present antigens, but they're technically antigen-presenting cells. So where do these cells come from? This next little section is about the origin of B and T lymphocytes, these cells of the adaptive immune system. They, of course, originate in the bone marrow. They are white blood cells, and all of the blood cells originate in the bone marrow from stem cells and progenitor cells that eventually differentiate into different white blood cells. And here we have the lymphocytic lineage producing B cells and T cells. The B lymphocytes, when they come out of the bone marrow, they've been matured, as we say, or educated, and they are ready to go to work. And T lymphocytes instead, though, go to the thymus gland. They travel in the blood to the thymus gland, where they will go through a maturation process. And then these cells will go out and seed, quote unquote, or live in lymphoid tissues throughout the body. We saw that lymphoid follicles are just packed with B lymphocytes, and they're found in other non-follicular lymphoid tissue as well. So lots of B cells live in the lymphoid tissue. T lymphocytes tend to rove through the body, but they're traveling through those lymphoid tissues as well. So that's what happens. These cells go out from their origins to populate the lymphoid organs so that we can have immune responses. And if they encounter an antigen, they will become activated and they will mete out an immune response. If a B lymphocyte, this little arrow suggests that this B lymphocyte is going to leave the lymph node. It's just trying to say inside this lymph node is a B lymphocyte that if it finds its antigen or encounters its antigen, it'll become activated and it will multiply and produce mature cells called plasma cells, which are actually where the antibodies will come from. So what does that lymphocyte education really mean? An education of lymphocytes or maturation of lymphocytes, it's not the same as a maturation, say, of a keratinocyte. It's a process of selection. And there's something really amazing and almost magical about the immune system, and that is that each lymphocyte has a unique receptor on its surface for an antigen. So every lymphocyte is unique and different. Its receptor looks like the receptors of the other lymphocytes, but on the tip, just like on an antibody, there's a recognition sequence that recognizes one specific or particular antigenic determinant. And there are billions. So the way they're produced is at random. There's some genetic shuffling that happens so that at random, all these B cells and T cells are produced, each with a unique receptor on their surface for every possible chemical substance as far as we know. So we start out with this incredible array of cells that could recognize anything, including the proteins of our own body. That's how they come out of the red bone marrow initially, or out of the sort of lineage process. And then we have to get rid of the cells that would recognize our own proteins. So in the thymus gland, for example, T lymphocytes, we have to carefully pick through all the T lymphocytes, and if any of them recognize a protein or a substance from our own body, we need to kill that cell and get rid of it and save only the ones that recognize therefore something foreign. By difference, we'll wind up with a whole selection of T cells whose receptors only would recognize something not found in our own body. That's called self-tolerance. So we're really just selecting out the T cells that are going to be useful for destroying foreign stuff that's not going to attack our own body. Well, there's actually kind of two steps to that, and it's because T lymphocytes, it turns out, can only recognize antigens when they're bound to a major histocompatibility complex on a cell. So the first thing we have to do in picking out the good T lymphocytes that are going to do the job that we want, we have to pick out ones that have a receptor that can successfully identify the MHC complex. And sometimes when the cells are made, they just don't come out right and they don't recognize the MHC, or they recognize it too avidly. So the first step in so-called education is positive selection, getting rid of all the T cells that were produced in the red bone marrow that don't properly recognize MHC. And then we, from that, go on to see which ones will only recognize a foreign antigen with their T cell receptor. So there's a receptor for the MHC, there's a receptor for antigens, and we want to find T cells that recognize MHC and then have a T cell receptor that recognizes only foreign agents in our body. That's the process of selection. Here's a cartoon form that illustrates this process. So here, this is the positive selection at the top half of this illustration. There's a T cell here, a developing T cell, and here's kind of like maybe a nanny cell in the thymus who's going to be holding one of many that are going to be holding antigens that represent antigens of our own body and that have MHC on their surface. So the first thing we're going to do is see if this T cell can recognize the MHC. Here's supposed to be a cartoon showing that this T cell didn't come out right. It doesn't recognize the MHC complex properly. We're going to get rid of that cell and induce apoptosis, cell death. Get rid of it. And here's a nice T cell whose CD4 or CD8 receptor can identify, can bind, we'll talk about those later, can recognize the MHC complex, and yes, we're going to keep that one, and send it down to the next station. And then in the next station, we're going to take a look and see whether its T cell receptor in the center here, the antigen detector, that's represented by this little kind of like rounded groove, recognizes a self-antigen. This nanny cell, remember, is holding a protein fragment from some part of our own body. If the T cell does recognize or stick too avidly to that self-antigen, we're gonna kill that T cell, get rid of that one. And here's one that the little hole is supposed to represent. This one just doesn't recognize that self-protein. It doesn't stick, it doesn't bind, and so that's a good one. So this bottom cell recognizes MHC and does not recognize our own body proteins, and now that's a keeper. So then these things go out into the lymphoid tissues and are roving throughout the body. They're called naive cells. They have been selected, but they have never encountered, the antigen is never actually bound to their receptor before. So we're going to send them out there to work, and they stay out in the body and wait for that moment when an infection occurs that they would recognize. So what actually happens? What's the process of activating an adaptive immune response? So when an antigen comes into the body, a humoral immune response is the first thing we're going to talk about. An antigen will be detected by a B lymphocyte, and it will start the process getting ready to produce antibodies. So we'll be talking about that first. If an antigen came into the body, an infection took place, and a T lymphocyte was the one that recognized the antigen because it was lurking inside of a cell, for example, then cellular immunity would undergo. And so we'll be talking about cellular immunity in the next section. And finally, in that second section, also we'll talk about helper T cells. Helper T cells are a type of T lymphocyte that's needed for either one of these reactions to take place. If a B cell finds its antigen without some help from a helper T cell, that's why it's called that, it won't produce any antibodies. It'll stop. So here are the steps of the process of activation and maturation and production of effector cells of the adaptive immune system. This list is common to both humoral and cellular immunity, so keep it in mind. We're just going to jump right into talking about humoral immunity, but keep in mind that T lymphocytes are going to follow the same set of processes in order to get to the point where they can do their job and defend us. So clonal selection, first thing we need to do is find a B cell who's appropriate for the job that needs to be done. Once we find a good B cell who can react to a particular infection that's in our body, then we're going to cause that cell to multiply or proliferate and form an army of identical cells that can then mature into what are called plasma cells. Plasma cells are specialized protein-making factories that can just churn out antibody proteins like nobody's business, and those will go on to protect us or defend us against the infection. But at the same time, here's something really important. At the same time that the plasma cells are being produced by maturation to make antibodies, some of the B lymphocytes that were produced in this clonal proliferation process will be turned into pre-programmed memory cells. So when we talk about immunological memory, we're talking about a population of cells that has been exposed to the antigen or is the product of a proliferation process because of a cell that was exposed to an antigen. And now you have a batch of cells that will stay in the body for years to come and be ready to do something new and different if that same antigen ever dares to come back on the scene. So clonal selection, we said that the process of making lymphocytes involves a genetic shuffling process. We have billions of lymphocytes, or at least a billion or so, individual, unique lymphocytes whose receptors recognize different, each one represents a different, recognizes a different antigen or antigenic determinants. We got this whole array of cells. Now an infection comes in the body and it has some foreign substances on its surface. We need to find a B cell whose receptor recognizes that particular antigen or the antigenic determinants. So that's the process of clonal selection. We got to shop around the antigen until it comes in contact with a B cell that does recognize it, and then that B cell will become activated. So that's the process. It's like the Cinderella and the glass slipper analogy. The prince had to travel all through the countryside trying to find the foot that would fit properly into that glass slipper. We're trying to find the B cell, or later on we'll talk about the T cell, whose antigen receptor matches the antigen that's in our body today. Once that happens, the B cell becomes activated, and if there's a signal present from a helper T cell saying, yes, this is actually an infection, we have some corroborating evidence that this is a real deal, and we do want you to go forward with this process, then that lymphocyte will proliferate. It'll just multiply by mitosis into a clone, or an army of identical cells, each one whose antigen receptor is identical to that one that we so carefully selected by clonal selection. Now we have an army of cells with the right kind of receptor, and it turns out that those cells will then differentiate into plasma cells that will produce antibodies, and the antibodies, importantly, will have recognition sites on those little Y-like tips that's identical to the cell receptor that we use to select that B cell in the first place. One interesting little analogy that's used sometimes to talk about this activation process and the second signal that's needed from a helper T cell. Think of the clonal selection process as like going out and starting your car. Get in there, put the keys in the ignition, start the car, or if it's a modern car, just press the button on the dashboard. It starts up and it's just sitting there, and you could leave it there and it would run all day until it ran out of gas. It wouldn't go anywhere at all. That's like a B cell that has been activated but not given that second signal. If you get into the car and put it in drive and begin to move around and go places, that represents, or is the analogy to the B cell that has received the second signal, will proliferate and produce a mature population of cells that can make antibodies, and importantly, some memory cells. Some of the cells that are produced by the differentiation process from the clone are going to stay in the body and wait for the possibility of re-exposure to the same antigen. So here's a little cartoon depicting all these steps. Here are some B cells, and they've drawn the B cells with different colored surface receptors. It turns out that the antigen receptors on a B cell are in fact just antibodies that have a special stem part that can attach onto the cell surface, and they've made them different colors to suggest that each one has a different antigen receptor that binds only to one particular unique antigenic determinant. And so these little red things represent antigens that are coming into the body on this particular day, and they bounce off this green B lymphocyte, they bounce off this light blue lymphocyte. They don't bind to the antigen receptor. The antigens actually stick on to this one because it has a B cell receptor that is specific for that antigen. Now that B cell becomes activated. As long as there's a second signal present, this cell, this middle cell, begins to multiply. We only have two here, but you have to use your imagination. We're going to form a whole army of identical lymphocytes that have the same B cell receptor, and then they will mature into plasma cells. These guys have a lot of endoplasmic reticulum and ribosomes. They can make protein. They're factories, and out comes antibodies that are identical to the B cell receptor in terms of antigen recognition, and they will go on and sweep those antigens from our body. We'll talk about that later. How do antibodies actually help us out in this way? That's a separate discussion for a different slide. Anyway, but it's important to remember that some of the cells produced during the proliferation process become memory cells. Memory isn't some magical ethereal thing like when you remember something from childhood. It's physical cells They're pre-programmed cells that are coming from this proliferation step. They've been clonally selected, activated, proliferated, and now we're just going to keep a few of them and save them in case we're exposed to the same antigen again. Something new and different is going to happen. So that is the process of humoral immunity. You've got the effector, it's called. The effector cell is a plasma cell, but the effector itself is the antibodies that are going to protect us from the antigens ultimately. T cells, as I said, are going to follow the same kind of clonal selection and activation and maturation process. It's just a little different in that case because T cells don't recognize antigens that are just floating in the lymph fluid, for example, or in the bloodstream or wherever. T cells only recognize antigens that are attached to MHC complexes on the surface of cells. So there's a little more to it. We have to figure out how that's going to work. But T cells, once they discover their antigen on an MHC, will become activated and proliferate and so forth. And eventually, the effector cells, in the case of cellular immunity, will be cytotoxic T cells. Cyto just means cell, and toxic, that can kill infected cells in your body and destroy them. And this little table just kind of summarizes what we're talking about with respect to the two, cellular and humoral immunity. And you can come back and refer to that at a later time because we haven't talked about cellular immunity yet. Let's look at another really, really important aspect of humoral immunity before we go on to talk about cellular. Immunological memory, as we said, is referring to the presence of memory B cells that are going to stay behind in the body after an immune response takes place. That immune response that we've been talking about so far is called a primary immune response. It's the first time that the adaptive immune system has been exposed to an antigen. The first time a B cell has been clonally selected for this antigen, and all the steps took place. We formed some antibodies, and now we have some memory B cells. They come from the primary immune response, first exposure. A secondary immune response occurs when the same antigen is presented to our memory cells. We get infected with the same cold viruses, for example, and that cold virus starts to attack our body, and those memory cells are there this time, and a much faster, more vigorous, more prolonged response takes place, and that cold virus will never have a chance to produce any disease symptoms, or it could be diphtheria, it could be measles. It will never have a chance to produce disease in our body because the memory cells will trigger such a fast and aggressive response, it'll be wiped out before it ever gets that far. So that's called a secondary immune response. It's the reaction of the memory cells to a re-exposure. And here it is in cartoon form, just to illustrate it. Here's the top part is this familiar thing we were just looking at, showing a primary immune response which culminates in producing antibodies, but importantly also memory B cells. And the bottom half is a secondary response in which the same antigen has come into the body again, right? Because it's present in the environment. It's something common. And it comes back into the body again, and the memory cells bind with their surface receptors, their B cell receptors, to this antigen, and bam, within just hours or a day, they produce a giant clone, a much bigger clone than before, differentiate into a huge population of plasma cells that can make an enormous amount of antibodies. So just everything happens bigger, faster, and we're just not going to let that antigen have a chance to infect our body this time. And even more memory cells are now produced. So we're turning into a super responder to a particular agent, particular antigen. Here's a graphical look at the concentration of the antibodies produced by this humoral immune response in a primary immune response and in a secondary immune response. The way we talk about concentration of antibodies when they're in the blood, that's often how we try to scope it out. We take a look at the blood and see how many antibodies are present. What's their concentration? That's called titer. You may have heard of antibody titer. It's just the concentration of effective antibodies in the blood. Okay, so we have exposure to an antigen. That's day 0, right? On the x-axis are days of time. And day 0, we get exposed to an antigen, a primary exposure. Primary immune response involves a lag of maybe as long as 6 days, even 7 or 8 days before the concentration of antibodies reaches an effective level in the blood. It takes multiple days to do clonal selection. activation, proliferation, maturation, starting to secrete enough antibodies that they accumulate. All those steps take time. But now we have memory cells left behind, and eventually the antibodies don't last forever in the circulation. They're there to wipe out the antigen, but then eventually they become kind of degraded. They become old and they're removed from the circulation, and fresh new proteins take their place. But if we get exposed to the same antigen again, this arrow represents a secondary exposure, a secondary immune response, and the memory cells are present. We have 0 days, 1 day, and we already have antibodies starting to be produced and come into the blood. And look at the titer. Within a couple of days, we have this enormous titer of antibodies that has been produced, and that's so fast that the pathogen, the disease-causing agent, is not even going to have a chance to produce any symptoms. We're going to destroy it. So that's the difference between a primary immune response, lag, and only a certain amount of antibody production, versus a secondary immune response, no lag, and a massive production of antibodies. This little pink graph line right here represents a control. Whenever you do an experiment, you need to have a negative control, and just to prove that this primary immune response didn't just rev up the entire batch of lymphocytes, rev up the entire immune system, here we have chosen to introduce a different antigen into the body at the same time as our secondary exposure. So antigen B was introduced here, and lo and behold, it still took a week for antigen B to drive a humoral immune response and produce antibodies. So everything is exactly the same in the body except the memory cells to that antigen A that made for that secondary immune response to be so swift and vigorous. So that's so cool. Why is that so cool? Because it has to do with immunization, for one thing. So now we're going to talk about active and passive humoral immunity, but we're also going to talk about naturally acquired versus artificially acquired immunity. So we're going to wind up with 4 categories: active and passive immunity, and in each case, natural and artificially acquired immunity. So what is active immunity? Active humoral immunity means that an antigen came in the body and a B cell was selected, and we had the whole thing happening, and we wind up with antibodies being produced and memory cells. So that everything we've been talking about so far is an active humoral immune response. And so usually what we're talking about is you get sick, you get an infection, and your immune system responds. We have clonal selection, activation, all those things happen. We get antibodies and we root this thing out of our body and we get better. But when we use vaccines, what we're doing is we're letting the vaccine take the place of the primary immune exposure. We introduce the vaccine, which has antigens from a pathogen, a disease-causing bacterium or virus, and introduce that into the body. It creates a primary immune response so that now we have memory cells against antigens of a very dangerous and terrible disease-causing nature, but we already have the memory cells without having experienced the actual disease infection. And so that when we have a secondary immune response in the case of vaccination, we'll never get the disease because, again, the memory cells trigger such a vigorous and swift immune response that will never get the disease. You don't get diphtheria, you don't get pertussis, you don't get who knows, pneumonia, because you've been immunized against these things, and you have memory cells in your body, so that if those things come into your body, they will be destroyed in no time at all. Passive humoral immunity. Sometimes time is of the essence and we want to get some antibodies into the body immediately. If someone is bitten by a rattlesnake, for example, and the venom is going to kill them in a matter of hours, we can take an antiserum, some serum, the plasma essentially from an animal that has antibodies against that venom, inject that right into someone's body. Those antibodies will latch onto the venom molecules and, as we said before, block them, and they'll be swept away by macrophages in the liver, and then the person will survive. So that's an example of why we want to use passive humoral immunity. It's great because we just saved the person's life. It's not so great in a way because we didn't produce any memory cells. The next time they get bit by the same kind of rattlesnake, they're just as likely to die from the venom as they were the first time. So that's the difference. Passive immunity is good for what it is, but it doesn't produce any humoral or any immunological memory. naturally acquired passive humoral immunity means immunoglobulins, antibodies that travel from the mother's blood through the placenta to the fetus. During pregnancy, all of the mother's antibodies, whenever she has an humoral immune response, her antibodies go right through the placenta, the circulation into the fetus. And when the baby is born, the baby is actually remarkably immunologically resilient. It has all the antibodies in its blood to all the stuff that's out there in the community that the mother has already been exposed to and made antibodies against. The problem is it has no immunological memory, so that eventually when those antibodies degrade, as we said, they don't stay around in the blood forever, they start to subside, and then the infant becomes very, very susceptible to virtually anything that comes along, and now it has to go through the sort of difficult process of producing humoral immune responses to every single thing that comes along and develop its own immunity, its own panoply of memory cells in its body. So that's called naturally acquired passive humoral immunity. And here's just a flowcharty box diagram of the same thing. So active immunity is actually exposing the immune system to an antigen, letting it do its thing. The whole process takes place. We produce antibodies by way of plasma cells and memory cells. It can either be naturally acquired when you get an infection, or we can vaccinate someone so that they will never have a disease when they get exposed to a particular antigen. Passive immunity means artificially injecting some antibodies into someone in order to protect them from something that we want to get out of their body, like right now, but they won't have any immunological memory, or antibodies from the mother's circulation that cross over the placenta into the fetal circulation that serve to protect that newborn for a while anyway, so that it can make it through those first weeks and grow and get strong and eat and so forth before it has to start dealing with antigens in the environment on its own. So the last section of this talk is about antibodies themselves. What are they? They're called immunoglobulins, and they come in multiple forms. There's different kinds of classes of immunoglobulins, IGs, and they all have the same Y-shaped structure with recognition sites. There's so-called variable chains that have the recognition sites. Variable means that every antibody is unique for a particular antigenic determinant. So every antibody has two recognition sites at least on that Y-shaped structure, and that's how it detects its antigen. The stems, the Y part, the stem of the Y part is different in each class of 5 classes of antibodies, and that's the different part. The Y parts are the same potentially, but the stems attach to different things and so forth give different functions to the different classes of antibodies. So let's take a look at the antibodies in the different classes. Here's an antibody molecule drawn in cartoon form. So there's 2 heavy chains, or long peptides. These sort of taffy-looking bars are chains of amino acids. It's a peptide. And so there's 2 heavy chains, and these yellow things are disulfide links. They're just side chains of a certain type of amino acid that can link together by sulfhydryl groups. And so these 2 different peptides, the 2 heavy chains are linked together, and then we have 2 light chains that are linked to the Y parts of the antibody, and together the light chain and heavy chain on the tips of the Y produce the specific unique recognition region of the antibody. That's how it recognizes its antigen. So there's 2 antigen sites on each antibody at least. Okay, so the stems are different, and in the case of immunoglobulin type M, IgM, it's a pentamer. It's just 5 IgG, or 5 Y-shaped antibodies, all made together into a pinwheel, and that's the kind of antibody structure that's first released by plasma cells. When you first develop a humoral immune response, immunoglobulin type M's will be produced by the plasma cells, these pinwheel-looking guys. It just produces a lot of different antigen recognition sites on each one that can cling to a lot of stuff, and we gotta, because we're just starting out, right? We're going out into battle. We want to have some major league good stuff going out there to start mopping up all the antigens. Immunoglobulin type A is actually a dimer. It's 2 antibodies whose stems kind of link together, and under certain conditions the plasma cells will be induced to produce immunoglobulin type A, which is secreted into mucus, saliva, other body fluids. So immunoglobulin type A will be found in body secretions. And again, it's an important defensive thing, but it just is very specific for where it will turn up in the body. So here's a little cartoon of an IgM, this pinwheel of 5 immunoglobulin, Y-type immunoglobulin structures in one. That's an IgM. Here's an IgA. It's a dimer, meaning 2 what would be Y-shaped antibodies stuck together at their stems. Immunoglobulin type D, the stem on that one just allows it to attach to the surface of a cell, and so a B cell receptor, the part of a B cell that allows it to recognize its unique antigen, is just an antibody with the stem that makes it immunoglobulin type D. So that's the B cell receptor. And then immunoglobulin type G is the main form of antibodies that are secreted by plasma cells. So plasma cells start out producing immunoglobulin M, and then after a period of time, they switch to making immunoglobulin type G. It's called class switching. Also, when there's a secondary immune response, when the memory cells encounter their same antigen again and they produce this whomping humoral immune response, they produce immunoglobulin type G typically. So therefore, immunoglobulin type G is what we find circulating in the plasma. The predominant immunoglobulin is type G. It's the common defensive immunoglobulin of our bodies. And incidentally, it can cross the placental barrier, as we said. Finally, immunoglobulin type E is like immunoglobulin G, except that its handle allows it to bind onto mast cells and basophils, the stem region. Once we have a humoral immune response in which immunoglobulin type E was produced, we may remove an infection from the body, root out the pathogens, but now we have antibodies that cling to the surface of mast cells, and then if we encounter that same antigen again, the mast cells are going to degranulate. When the antibodies on their surface bind to that antigen, they're going to degranulate. I use that term a lot. It means release their vesicles of chemicals, release histamine. We're going to have an allergic response. The antigen comes in and we have this whomping extraordinary inflammatory responses because those IgEs are on the surface of the mast cells, and out comes all that histamine. And so here again, just cartoon drawings of IgGs, IgDs, the B cell receptor. IgGs just don't have a cell binding region on their stem, and they just float in the plasma and so forth. And IgEs, which allow them to attach, has a stem that allows them to attach onto mast cells. We've already talked about class switching. Last section of this talk is to quickly identify. We know about how antibodies are formed. We know how the humoral immune response works. We know primary and secondary responses, why vaccines protect us from ever getting diseases for a particular antigen. What in the world do antibodies do to protect us from antigens? How do they save us? We haven't talked anything about the good stuff that antibodies do for us. So we're going to now look at a cartoon slide that will show us some processes that antibodies undergo to help us survive an antigen attack. Neutralization, we talked about blocking up an antigen. If an antibody binds to an antigen, now it can't do something else. It can't bind to a receptor on a cell, for example, because it's blocked, it's covered up, it's got some big chunk of antibody stuck to it. That's called neutralization. Agglutination, well, I'll go back real quick. Actually, sometimes bacteria, the problem that they produce in the body is that they produce toxins that can kill certain types of cells in our body. There's all kinds of toxins that do everything from harming your heart to killing epithelial cells to killing white blood cells that help the bacteria cause disease. When antibodies bind to those toxins, they can no longer attach to a cell receptor and cause those poisonous responses. Agglutination is really just something, it's talking about a response of your body if you are transfused with the wrong blood type to which you have antibodies, and there's an antibody reaction with this transfused blood. What will happen is the antibodies, those anti-A or B antibodies that are in your circulation, and they recognize those antigens on the blood cells that were erroneously put in your body, they will clump together large numbers of red blood cells because each antibody has 2 binding sites, so each one can bind to a separate blood cell, and then another antibody can bind to the blood cell onto another blood cell, and so forth. We form these huge aggregates of blood cells. That's called agglutination. But it really isn't a physiological thing. We don't physiologically become exposed to transfusion. That's a medical activity. But precipitation is essentially the identical thing. If there's a soluble antigen, a toxin, or a chunk of a bacterium that's been killed in battle and it's floating in the lymph fluid, that antigen can bind to an antibody, which can then bind to another antigen, which can then bind to another antibody, and so forth, and we can form big clumps of soluble antigens, and that can be a very important and powerful function of the humoral immune response called precipitation. Just means that precipitation means something that was soluble is no longer soluble. It comes out of the solution. It's no longer in the water. It's in a big glob, and it allows hundreds or thousands of antigens to be engulfed and phagocytized by Kupffer cells, say in the liver, instead of one at a time, and it just blocks and keeps things from spreading and getting them out and gets them out of our body faster. We know that the classical pathway of complement activation, this important process of lysing, if live bacteria get into the body somewhere in the circulation or in an area where there's an injury, the complement system can lyse and kill those bacteria and do all kinds of other good things. Well, complement activation is by way of antibody-antigen complexes. Antibodies bind to the surface of the bacterium. That causes complement factor C to become cleaved, and that starts the whole process of complement activation. So here's a cartoon showing neutralization. If this is a virus particle that could bind to the surface of a cell, get internalized and start a viral infection inside that cell, not anymore. Antibodies are going to neutralize that thing. It has been covered. It can't bind to the cell. Precipitation, we see a whole bunch of soluble antigens floating in the lymph fluid, for example, or in the blood, or any number of places. Antibodies bind to it and form this big lattice, and that will no longer be soluble. It can't go anywhere, and now it's going to be gobbled up by phagocytes. Phagocytes, macrophages, and so forth will eat up immune complexes and destroy whatever is attached to the antibodies. And so we're getting these things out of the body, getting rid of them by this phagocytosis and killing process. Incidentally, when we activate complement by antibody-antigen complexes, that activates inflammation in a big way. That helps us rev up the activity of phagocytes, call into the site a lot more white blood cells to help with the battle against infection. So we see there's a connection back and forth between the innate immune system and the adaptive immune system. They are connected in an important way, and here we see that one of those connections. So that's the way that antibodies can help us. Antibodies just coated on the surface of a bacterium, for example, as we talked about in the case of complement system, if antibodies attach themselves to an antigen, that just makes it tastier. And what we really mean by that is there's a separate receptor on phagocytes for the stem of an antibody. It's called the Fc region of the antibody. The Fc receptor on the macrophage will grab that thing by the stem and gobble it up. Just makes it that much more likely that the macrophage is going to get that thing out of the circulation. So that's our story. Thanks for sticking with me through a long discussion or description of humoral immunity and how antibodies can help us survive an attack by a pathogen. And the next time we meet, we'll talk about cellular immunity. How does the cellular immune system find, locate, and destroy infected cells in their body so that antigens can't lurk and hide themselves away inside of cells and escape from detection like they would if all we had was humoral immunity.